Hey friends, welcome to this channel. If you're new here, I am Indrani from Bong Biology, and in today's video I will be talking about the protein sorting via endoplasmic reticulum. To limit the duration of this video, I will make parts of the total process and will rename it as part 1, 2, 3 so that you don't get confused. So let's start. The endoplasmic reticulum is a network of membrane-enclosed tubules and sacs that extends from the nuclear membrane through the cytoplasm. We can say that this endoplasmic reticulum is the largest organelle of most of the eukaryotic cells. And depending upon whether the ribosomes are present on endoplasmic reticulum or not, we can divide this endoplasmic reticulum into three different types. One is RDR, SER, and TER. This rough endoplasmic reticulum or RER is covered by ribosomes on its outer surface. The smooth endoplasmic reticulum is not associated with the ribosomes, and TER is transitional endoplasmic reticulum where vesicles exit to the Golgi apparatus. Now both these RER and TER, they are associated with the protein sorting and protein processing, whereas the smooth endoplasmic reticulum is not associated with protein processing, but it is associated with the lipid sorting and metabolism. Now, the role of endoplasmic reticulum in protein processing and sorting was first demonstrated by George Palade and his colleagues in 1960. These investigators studied the fate of newly synthesized proteins in specialized cells of pancreas, that is, we call them as pancreatic acinar cells, which secretes digestive enzymes into small intestine. Because most of the proteins synthesized by these cells are secreted, Palade and co-workers were able to study the pathway taken by secreted proteins by simply labeling the newly synthesized proteins with radioactive amino acids and chasing them through autoradiography. So what they found is that the secretory proteins take a specific pathway to reach to their final destination, and that is called the secretory pathway. And that secretory pathway is from rough endoplasmic reticulum, they travel to Golgi apparatus. From Golgi apparatus, they travel to secretory vesicles, and from secretory vesicles, they travel to exterior of the cell. So, from rough endoplasmic reticulum, they will travel to Golgi apparatus. From Golgi, they will travel to secretory vesicle, and from secretory vesicle, they will travel to outside of the cell. So, this is called the secretory pathway, and these vesicles, they are being originated from these Golgi apparatus called the secretory vesicles. Next, we'll discuss about targeting of proteins to the endoplasmic reticulum. So, in higher eukaryotic cells, the initial sorting of proteins to the endoplasmic reticulum takes place while translation is still in progress. That means co-translational translocation. Proteins synthesized on the free ribosomes, that means free cytosolic ribosomes, they can either remain in the cytosol or are transported to nucleus, mitochondria, chloroplast, or peroxisome. In contrast, proteins, those are synthesized on the membrane-bound ribosomes, they are translocated either into endoplasmic reticulum, where their translation is still in process, that means co-translational translocation, and then they may either retained within the endoplasmic reticulum or are transported to Golgi apparatus, endosome, lysosome, secretory vesicles, and plasma membrane. And finally, these secretory vesicles, they will get fused with the plasma membrane, and then all the contents of the secretory vesicle will reach to the exterior of the cell. So now this is the incorporation of secretory protein into microsome. So I think you all know what microsome is. Microsome is, if we disrupt a cell, then the smooth endoplasmic reticulum and rough endoplasmic reticulum will be degraded and they will become small spherical ball-like structure. So these are called actually microsomes. Microsome produced from rough endoplasmic reticulum is called rough microsomes, and microsomes produced from smooth endoplasmic reticulum is called smooth microsomes. So what happened over here is the secretory proteins which are targeted to the endoplasmic reticulum is must contain a signal peptide or a signal sequence, this red color part. So this is the signal sequence. This is always present at the amino terminus end, and this signal sequence is removed during the incorporation of the growing polypeptide chain into endoplasmic reticulum. This thing was demonstrated by experiments showing that translation of secretory protein mRNAs on the free ribosomes yielded protein that retained their signal sequences and were therefore slightly larger than the normal secreted proteins. However, when microsomes were added to the system, the growing polypeptide chains were incorporated into the microsomes and the signal sequence were removed by proteolytic cleavage. So this signal sequence should be removed always. It should be removed before the protein is translocated completely within the microsome or within the lumen of the endoplasmic reticulum. Now we'll discuss something about the signal sequence. The signal sequence is a very short stretch of amino acid, around 20 to 25 amino acid, and 7 amino acid among this 20 to 25 amino acid should be hydrophobic. Okay, and they will be always present at the N terminus end of the protein. Okay, so this is signal sequence, and this is the main characteristic feature of all the secretory proteins. Hope you understand. So in next video we'll discuss how these secretory proteins get involved or get translocated on the endoplasmic reticulum membrane. Hope you enjoyed this video. Thank you.